We've spent this semester talking about animals, major groups of animals, in particular the chordates, the vertebrates, which are the major part of the chordates, and the arthropods. And we've given you an overview of some of the evolutionary changes that have resulted in major shifts in development and thus given different animal form. Largely evo-devo for the first dozen years, maybe 15 years, was all animals. And again, arthropods kind of the workhorse because they're diverse, they're easy to keep in the lab, there's a lot of really cool things you can study as far as particular changes. So they are a really great organism. Animals in general have the vast majority of morphological diversity. However, recently there's been a surge in the amount of data and our understanding of plant evolution and development. And so even though I'm an animal guy, I admit I'm a little bit biased. And animals, I think you can make a really strong case, are really the best group when we're looking at the evolution of form and diversity and where new things come from. We should not just ignore plants altogether. So they are important and we should look at them. This is going to be a rather short overview because it turns out that the principles of development are largely the same in plants as they are in animals. Now, they're incredibly distantly related. Now, all organisms on Earth do share a common ancestor, but the separation of the lineage leading to plants and the one leading to animals was a couple billion years ago. And so this is a huge amount of difference. And so the underlying architecture is very, very similar. The same basic trends, we'll look at a few differences apply, but the genes that are doing it are vastly different. Okay, other than being the same general class of genes, that they bind to DNA and turn on and turn off transcription factors, or they are transcription factors that turn on and turn off target genes. The genes themselves are vastly different. So no Nox genes, but there are genes with DNA binding motifs that serve somewhat similar purposes. Okay, so this outline here just says at the beginning many of the mechanisms are the same. But let's look at some major differences that are either being studied or still need to be understood when we're looking at plants and animals. And we're going to look at both some kind of life cycle general biology differences and also some of the physical and developmental differences that distinguish animals and plants. And you already know many of the basic differences, right? You learn about plants and animals in Bio 1, you define all these different eukaryotic groups and you learn some of the major features of both. And you already have an understanding. Some of these you might be familiar with, others you might have only brushed over or maybe even never really covered. So number one, there is a distinct alteration of generations at the origin of all plants. And although it's not as clear in our larger plants because one of these generations, the sporophyte, is largely contained in the seed and is very small, and we really don't see it. It doesn't have a large physical presence in the higher plants, but it is still there. But in the early plants and many of the lineages that lead to our modern angiosperms, they still have this very clear alteration of generations, a distinction. That part of the life cycle is diploid. They undergo meiosis and they go and they make these haploid cells. These haploid cells will then go through mitosis and make two different types. And in the early plants, they are distinguished, but they're not like modern pollen or ovules, but they would basically make the equivalent in animals of eggs and sperm, the gametes. And this is why many of these early plants require water, because these are flagellated cells that need to fertilize. Later, they lose their flagella and they become transported via other mechanisms. But early on, these were flagellated cells that would then fuse together to, again, recreate a diploid organism, and the whole process would start over. And this haploid organism, in ferns, you can see it. If you know ferns well, you'll see it. It looks quite different from the fiddleheads and fronds that we typically associate with ferns. And it is the reproductive stage. Now, the ferns make these little haploid cells. We often call them spores. And if you look at a mature fern, often on the ventral, the underside, not ventral, that's an animal term, but on the underside of the leaves, you'll see these little brown clusters, which are called spores and are those haploid cells that will go out and form these haploid part of the life cycle. Okay, so just be aware, be clear that that was a very early difference, and although that's been modified so it's not as obvious in modern plants, all plants to some extent still have that, and it's very different than animals where there really is no haploid stage other than the eggs and the sperm, right? So imagine if an animal created a little spore and it went out and then created a new animal, quite different looking from the other one, and then those would make the eggs and the sperm, they would get together to then make the whole thing start over again. That's the way plants do it. So that's one major difference. Number two, there is no clear separation in plants between the soma, which means body, right? The physical, the main portion of the organism, and the germline, which are the cells that lead to the gametes. So in most animals, but not all, there is a very, very clear separation, right? You can't chop off an arm and have it grow up to be a little person, right? Or a tail from a cat doesn't have the ability to switch over and grow legs and a head and then eventually break off from the main cat and run away. But plants can do that. So there are many, many forms of vegetative growth and vegetative reproduction that are possible in plants. This can happen through root systems. It can happen from buds coming off of the major one. It can happen through mechanical damage where you separate them. And some plants actually can only reproduce asexually. And so it's quite easy to take the soma, right, the body of a plant, a stem, a branch, and relatively easy. Some plants it's harder than others, but turn it into roots. So you can take a cutting from a plant, and especially with a little bit of hormone, but sometimes even without it, you can put it into some moist soil and get roots to grow out of the stem. And it can be any part of the stem, although some work a little bit better than others. If you cut it right below a node and have some buds that are growing up, those tend to work a little better. Okay, so just be aware that the gametes, although they are still there and present for reproduction, there's a lot more flexibility with the soma, and soma can turn into plants, and then that plant would grow, and eventually, if it was a flowering plant, it would make flowers, and so that little bit of the root would turn into gametes. And there is no clear set of cells set up early in development, like in animals, that is going to just be the gametes, right? Gamete development can be initiated just about anywhere along the soma, depending on the needs of the plant. Number three, they have this interesting kind of triploid food source. So basically, when pollen fertilizes an ovule in a plant, there are actually two fertilizations that take place. So we have the original embryo that forms from a single gamete from the mother and a single gamete from the father. But then there's another one that forms from two gametes from one, I think it's the pollen that has an extra one. There are actually 3 nuclei in the pollen. And they both fertilize another one, and that makes a triploid protein oil-rich food source. And humans take advantage of this. So when you look at corn, beans, or peas, or any of the other organisms where we harvest the seeds to eat, we see the little tiny embryo. You can see with this next time you eat a peanut. Open it up and you can see this. This little tiny thing here is the embryo. And all the stuff around it is this triploid food source. Okay? So just be aware, and they've labeled the different parts of the developing embryo, that is a very clear difference. It's almost like a separate fertilization event, but it only creates a food source, not a viable living organism. And in plants, we can see this in animals also. There's imprinting, which is involved in the development of this food source. So all you really need to know is the definition of imprinting. It's a genetics thing, a little bit side topic, but an important difference. And this is common in all plants, and some animals will do this to some extent. And we're just beginning to understand the range and importance of this in life on Earth. But imprinting is when alleles that are inherited from the parents are not expressed. And so basically we can turn off some of the alleles. And how that's decided and which alleles are used and which alleles are silenced can be well, we don't understand it in a lot of detail for all ways, but there are various ways that can happen. So just be aware of that, and with plants that happens very, very commonly. And that is one of the reasons why plants are so immune to, immune is maybe not the right word, but it's easy to have a genome duplication in plants and not have it be detrimental to the organism. In animals, if you get a genome duplication, so you double your number of genes, usually that animal is dead. It's a very bad major phenotype. And it has happened a couple times, or at least large portions of the genome have happened, but it's a kind of a lightning in a bottle, a rare thing for it to work with animals. But with plants, genomes duplicate all the time and have been a major force in the evolution of new lineages and new species of plants, because if you have 4 copies of your gene, you're just turning off 3 instead of 1, and you still end up with this very similar phenotype. Or at least that it does make differences. That's why it's important for evolution, but not so different that it's likely to result in a very detrimental negative phenotype. Okay? So let's look at the history of plants. You already should know a little bit of this from evolution. We do a very little quick run through it. You've learned about it in Bio 2, I believe, is where they cover this also. But these are the things that we're interested in. We're interested in the events, but from an evo-devo perspective, the structures, the cells, the new features that allowed plants to make this transition into a new environment or to do something in a way that their ancestors never did. Okay? So number 1 is this major thing early in the history of plants was this transition to land, right? And this resulted in increased complexity of structures. We need support structures. We need vascular systems to move both food from the leaves down to the rest of the plant, and water and minerals from the soil, right? The roots pick that up and move that up. So we have to have all this increased complexity if we're going to survive on land. And there's a nice stepwise procedure to how this occurred, and many of the steps along the way still have extant relatives. So we have early mosses and liverworts that are the descendants with many of the same features as those ancestral forms that first transitioned on land. And then we have lycophytes, which are the early kind of vascular plants with very simple vascular tissues, and there's still some of those around. And ferns, which is the next step along the way. And then we have the conifers, right? And so conifers have seeds, and we start to develop these seed plants, but we don't have flowers. And then finally, the angiosperms. Now that's a little bit of an abbreviation, but if you remember those 4 major groups, you've got most of plants. Maybe it's 5. Let's review. So we've got mosses and liverworts. We've got the lycophytes. We have the ferns. That's number 3. We then have the gymnosperms or conifers, right? The plants with seeds but no flowers. And then we have the angiosperms, which represent the vast majority of plant diversity on life on Earth. So angiosperms are the plants with flowers. So 5 groups, right? So just be aware of those. And at each of those groups, there are major innovations that are really kind of changing for plants that make a dramatic, major difference. And there are many other changes also, but those 5 represent major innovations and gains of new features for the plants. And so there's a lot of work and ongoing work to develop new models and study organisms that span the phylogenetic changes between those major groups. And one of the things that's been discovered as we have development of this more morphologically complex groups, that we do see increases in the genes. Now we're not going to memorize where and when, and they're not as obvious as the Hox gene transitions that we talked about with animals, but there are some very clear genes. And one of the groups of genes that was critical during the evolution of some of these complex forms are called the Nox genes. It's kind of a takeoff, a riff. They were named to sound similar to the Hox gene. So important developmental genes. They are transcription factors, but even though the name is similar, they don't seem to have a common origin. They're not paralogous copies of the Hox genes from the animals or anything like that. So they are really kind of separate and unique and distinct for plants. And we'll talk about one other group of genes in plants, but we don't really have the time or inclination to get into lots of detail. So you should know the Nox genes, and that they were just an important gene diversification that allowed for the morphological diversification of the early land plants. Okay? And so we can even call them homeodomain genes, and we see some of the same binding patterns that we see in these other transcription factors in the animals. Okay, number 2. Next major step after transitioning to land was the evolution of the leaf. And in fact, as we map it onto the phylogeny, the leaf, or at least leaf-like structures, seems to have evolved multiple times and then been modified greatly in many different groups. So conifers may represent a separate leaf origin from the angiosperms. Cactus don't have leaves, most species of cactus. However, they do, when you look developmentally, have an analog which has just been modified so much that we don't recognize it, and those are the spines. So it turns out cactus spines are modified leaves, and we know that both from looking at the physical development of those spines, and also now as we begin to identify genes that control the development of the leaf, we look at it and see very similar things. And then of course shape, variety, diversity, color, all of the different things that we see in leaves. This has been a critical breakthrough in the evolution of plants. So to make a leaf, there are a couple of things that we need to do. And again, we won't get into the genes for this, but just be aware that these are the things that happen. We have axes that need to be patterned, much like in in animals. Now in animals, at least in the bilaterians, we have these 3 major axes that all bilaterians are built on, and we learn those. You can review those if you want. Plants really don't have that, right? They don't have a proximal and a distal side. We could define the trunk, but often then trunks will just sprout out of trunks, and so we have a second proximal distal axis, that's on the plant. So some portions of the plant do look like they are radially symmetrical. And radial symmetry, which we haven't really defined in class, but it's symmetry like in the jellyfish, the cnidarians, or in the echinoderms, which are secondarily 5-fold radially symmetric. Okay, so radial symmetry, I always think of it like a pie. You can cut a pie in half and have roughly even halves, and you can, no matter what angle you do that at, as long as you go right through the middle, you still have even halves. So think about a cross-section of a stem, or even certain leaves, where if you cut them, one side, one half, looks basically a lot like the other. And many leaves even have this kind of weird not weird, but distinct, different from animals. And they have a bilateral symmetry where one half of the leaf looks very much like the other half of the leaf. And that's true for many leaves, but not all of them. Okay? So axes do get established, but they are more varied, and we don't have an overall 3-axis plan upon which plants are built. So we do have axes, but they're varied depending on the structure and may not apply across as wide a taxonomic range as the ones we looked about in animals. And the Nox genes are also involved in flower development. In leaf development. We'll talk about flower development in a little bit. So these Nox genes, which allow the transition to land, are also then further diversified and help to create some of the diversity that we see across leaves. So again, Nox genes, nice, easy name to remember since we've already talked a lot about Hox genes. And this process is very, very flexible, as is much of plant development. So think about it. If you look at a human, there's some very basic fundamental things that you can say, this is what, unless there's a mutation or an injury, this is what the human is going to look like. You know how many legs they're going to have. You know how many arms they're going to have. You know roughly where the heart is going to be. And although there is variability, the main body plan, right, the blueprint upon which all animals really are built, is very, very conserved. And so one human within a fairly narrow range has all of the morphological features that another human does. But plants are quite different, right? So we sometimes see 2 main trunks coming up. So where does the main trunk branch? Does it branch in 3? Does it branch in 2? Which direction do those go in? Do they go opposite one another, or do they grow alongside? And so for plants, this is incredibly labile and flexible. Okay, so labile just means easy to change and maneuver, but still maintain integrity of the system. Now, finally in the angiosperms, right? So we kind of skipped over conifers, but there's some interesting things there. We can look at the evolution of the seed, the paper that I've put online, and by the way, I've also put in that same folder a review, some guideline questions, rather simple and straightforward that you should be able to answer and are fair game for the test. It's a good short paper, so a lot of the papers were complex and long, and I didn't have you read them because we just kind of grabbed the major points in lectures and talked about those. But you should read paper 13. It's only a page and a half long, and then also make sure you go over those review questions and make sure that you can answer them. Well, the next major event is the origin of the angiosperms, the flowering plants. And it turns out that when we look developmentally and look at the genes that cause early development, that a flower is actually just a highly modified ornate leaf, or maybe many leaves put together. So the same things, the same genes and the same developmental pathways that grow a leaf were co-opted and then made more complex. So we ended up with flowers. And this is actually a flower that has been modified genetically so that the normal growth patterns are gone, and we just have a repeated, I think it looks like a stamen, repeated over and over and over again. So the flower is a modified leaves, and that means each of the part of the flower we can look at as a leaf that has been modified for a new function. And there's been a ton of work that's gone into the flower, both in the diversity and where we come from, but originally just kind of the fundamental question of where do flowers come from. Now, the vast majority of the work that has been done on this, and in fact most plant evo-devo questions, is done with Arabidopsis. And Arabidopsis is a member of the mustard family. It's an almost perfect model plant because it's small, easy to grow, grows like crazy, makes the thousands upon thousands of little tiny seeds. So it's a good model organism, and it is basically the plant equivalent of the fruit fly. However, if we want to study and understand other systems, we may not be able to use Arabidopsis, right? So if we want to look at the origin of the seed, Arabidopsis may not be the best plant. It gives us a framework for, okay, maybe this is the way angiosperms do it. But we may want to also develop other lineages into model or near model systems, so we can look at different groups that span these interesting evolutionary events. And so the paper talks about briefly about a fern model that's been worked on and evaluated, and that becomes a basis to begin almost a null hypothesis for the evolution of the seed, because they don't do it. So we can look at the genes that are controlling the seed development in seeded plants, and then look at what's going on in the ferns. So Arabidopsis still is the workhorse, but more models are needed and being developed. And the paper makes this a really clear point and spends a couple of paragraphs looking at some of the recent work in the model or near model systems that have been developed. But it's really something, and I've made this point in class also, that applies to all organisms. A model approach, model system approach, can only take you so far. At some point, you need to be able to look at a range of organisms, see if they all do it the same way, and then you say, oh, this is a fundamental feature of animals or of plants. But especially where there are major morphological differences, you need to look at 2 or 3 or 4 to be clear about where those changes occurred and whether those changes happened once or happened multiple times. You can do that by looking at a range of organisms and understanding them in more detail. So we are going to finish up by looking at the model that is the current understanding for the origin of the flower. Number 1, it's a modified leaf or group of leaves. Number 2, there are 5 genes that control its development, and these have been called A, B, C, D, and E, and they are known by other names also. And in fact, originally there were just 3, and then later the others were discovered, and it gets a little bit more complex. But it illustrates something that we've already talked about. So in the center of the tissue where the flower is going to develop, these genes are made, and some of them are in the middle and only located in the middle. Others diffuse out and make like a ring, like a bullseye pattern. So you can see it here on the left, where B is like the middle ring in the bullseye, A is the outer ring, and they overlap, and then C is this area in the middle. Now, notice here that they've really only drawn like the left-hand half of this. They put B on the bottom instead of on the top like in this one. But they've only drawn the left-hand side of this whorl pattern, this bullseye pattern. We won't worry about D and E. Those were found later and are involved in more complex flower morphologies. Okay, but A, B, and C in Arabidopsis, A, if it's all by itself, controls sepal development. Sepal are those green, they look like green petals, but they're on the outside of the developing bud, and then as the flower opens, they are at the bottom there. If you have A and B overlapping, so where they are both expressed, you grow petals. Then B and C overlapping, you grow the stamens, right? Those are the male reproductive parts. And then the carpels, the female reproductive parts, are only where C is, so right in the middle. And so you get and this is the earliest flowers, and many flowers have a very similar or a somewhat similar form, but others have modified variations and differences. So in the very middle, one gene, you don't really need to know which one, but one gene grows the female reproductive parts right in the middle of the bullseye. That gene, when it overlaps with B, grows the male reproductive parts, and most flowers, they have both male and female reproductive parts. Right? There are some where it varies, where we have male flowers, which only make pollen, and female flowers, which only make the ovules, the female gamete. Okay? But most of them make both. They're hermaphrodite reproductive structures, basically. Okay? Dioecious. Dioecious is when you have a plant that is male and then a plant that is female. So most of them are really kind of hermaphrodites. And so this is called the A, B, C, D, E, after the names for those 5 genes. It's an important model for you really could say the most important innovation in modern plants. The vast majority of plant species where all of the biodiversity is are flowering plants, and they have the most morphological diversity, they are most widespread and live in the most variety of ecological conditions. So just review that, make sure you understand, especially the ABC one. And there's this interesting question. It doesn't go into a lot of detail, but it gives a pretty clear answer in the paper about this ABCDE model. And it turns out that as we have expanded our sampling of flowering plants, that not all plants have that A gene to control sepal development. So that may be something that is derived and unique to a small group of flowering plants, of which Arabidopsis is part of. And so it can be tricky to understand and say, oh look, Arabidopsis is this great model, here's how we make flowers. But you don't know what features of those may be newly evolved and changed from the ancestral form until you can put them in their phylogenetic context and expand your sampling beyond just the supermodels, the main model of evolution.